Did you know that chronic inflammation not only causes pain or immune problems, but is also a breeding ground for cancer? Today, we are going to reveal a key truth. Can we really stop the migration and spread of cancer cells by regulating inflammation? In this video, we will take you to learn more about 5 anti-inflammatory nutrients supported by scientific research. They are not only safe, but may also become a natural barrier against cancer metastasis. Part 1. Inflammation is the driving force behind cancer. Chronic inflammation runs through all stages of tumor development. 1. Initiation. DNA damage caused by reactive oxygen loss and reactive nitrogen RNS. Free radicals attack genes and cause mutations. Reactive oxygen loss are oxygen-containing free radicals oxides, such as superoxide anions O, hydroxyl free radicals O, hydrogen peroxide HO, can trigger oxidation reactions. Reactive nitrogen RNS are nitrogen-containing free radicals nitrides, such as nitric oxide NO, peroxynitrite ONOO. Can trigger nitration nitrosation stress reactions. 2. Promotion stage promotion. Secretion of cytokines such as TNF-alpha and IL-6 promote cancer cell proliferation and invasion. Cytokines are a class of small molecule proteins secreted by immune cells, tumor cells, endothelial cells, etc., which have signal transduction functions. They play a key role in regulating immune response, inflammation, cell growth and migration. TNF-alpha tumor necrosis factor alpha, secretion source, macrophages, T cells, tumor cells, etc. Function, inflammation initiation, cell apoptosis, and metastasis promotion. Role in cancer cell migration. Activate NF-kappa B, MAP, JNK, and other signal pathways. Promote EMT, epithelial-mesenchymal transformation. Improve cell migration and invasion ability, and activate MMP9 at the same time. IL-6 interleukin-6, secretion source, macrophages, adipocytes, tumor cells, etc. Function, inflammation maintenance, growth promotion, and metastasis promotion. Role in cancer cell migration. Activate JAK, STAT3, PI3K, AKT and other pathways. Promote cancer cell survival, drug resistance, angiogenesis and enhanced migration ability, highly related to metastasis. TNF-alpha is the igniter, and IL-6 is the fuel of the flame. The two work together to not only create an inflammatory storm, but also open a channel to the whole body for cancer cells. 3. Progression. Activate the NF-kappa B, COX2, and STAT3 signaling pathways to induce EMT epithelial-mesenchymal transformation, causing cancer cell migration and metastasis. 4. Decompose matrix proteins MMPs to open channels, and cancer cells migrate throughout the body. Part 2. Systematic explanation of signaling pathways and mechanisms related to cancer cell migration. 1. The NF-kappa B pathway, nuclear factor kappa B, nuclear factor kappa light chain enhancer of activated B cells, is a key signaling pathway that regulates multiple physiological and pathological processes, such as immune response, inflammation, apoptosis, and cell proliferation. It plays a core role in diseases such as cancer, chronic inflammation, autoimmune diseases, and viral infections, and is an important pathogenesis of many diseases. 2. MAPK, mitogen-activated protein kinase, is a highly conserved signal transduction pathway that is widely present in eukaryotic cells. It is responsible for transmitting extracellular signals, such as growth factors, bacterial toxins, inflammatory factors, and stress, to the cell nucleus, regulating cell proliferation, differentiation, survival, inflammation, and apoptosis. Different mapped pathways are both independent and cross-linked, and are the basis for understanding cancer, inflammation, aging, and stress response. 3. The JNK pathway, C-Jun N-terminal kinase pathway, is one of the 3 major branches of the MAPK, mutagen-activated protein kinase signaling pathway. It plays a vital role in the process of cell response to stress, inflammation, cytokines, ultraviolet radiation, and apoptosis. JNK is particularly important in mediating cell death, apoptosis, immune response, and inflammation. 4. EMT, epithelial-mesenchymal transition, is a highly plastic and reversible biological process, which refers to the process in which epithelial cells lose cell polarity and intercellular connections, and acquire the characteristics of mesenchymal cells, i.e., fibroblast-like cells. This process plays an important role in physiological and pathological processes such as embryonic development, tissue repair, tumor metastasis, and fibrosis. 5. MMP9, matrix metalloproteinase 9, full name, matrix metalloproteinase 9, is an important member of the MMP family. MMP9 is a proteolytic enzyme that depends on zinc ion, Zn activity. Its main function is to degrade collagen, gelatin, elastin and other components in the extracellular matrix. It plays a key role in various physiological and pathological processes such as extracellular matrix, ECM, degradation, tissue remodeling, inflammation, tumor invasion and metastasis. 6. JAK, STAT, Janus kinase, signal transducer and activator of transcription, is a signal transduction pathway unique to cytokine receptors on the cell membrane surface, which is used to quickly transmit extracellular signals from the membrane to the cell nucleus and regulate specific gene expression. Among them, STAT3 is the most valuable transcription factor, especially playing a key role in tumors, inflammation, and immune escape. 7. The PI3K, AKT pathway is one of the main pathways for cell growth, survival, metabolism and anti-apoptosis, and is controlled by multiple stimuli such as growth factors, insulin, and cytokines. This pathway is highly active in cancer, diabetes, and neurodegenerative diseases. 8. COX2, cyclooxygenase 2, is an inducible enzyme that plays a key role in inflammation, pain, fever, tumorigenesis and progression. It is the rate-limiting enzyme for prostaglandin synthesis and is involved in the process of converting arachidonic acid into prostaglandins, PGs. High expression is closely related to the occurrence and invasion of various cancers, such as breast cancer, colon cancer, and prostate cancer. Part 3. 5 natural anti-inflammatory factors, key pathways to block the spread of cancer. 1. Omega-3 fatty acids, a class of unsaturated fatty acids that are very important to human health and belong to polyunsaturated fatty acids, PUFAs. They play an important role in maintaining cell membrane structure, regulating inflammatory responses, cardiovascular health, brain function, and vision development. It can inhibit NF-kappa B, IL-1 beta, and MMPs. It can significantly inhibit metastasis in colon cancer and breast cancer models. The American Heart Association, AHA, recommends eating fatty fish twice a week, which is equivalent to about 501,000 milligrams day. 2. Curcumin. It is a natural polyphenol compound extracted from the rhizome of turmeric, scientific name Curcuma longa. It is its main active ingredient, and the reason why turmeric is yellow. It is widely studied and applied for its anti-inflammatory, antioxidant, anti-cancer, and immune regulation activities. It can inhibit COX2, IL-6, STAT3, and NF-kappa B. More than 20 cancer models have shown that it can reverse EMT. Recommended dose, 501,000 milligrams day. Auxiliary for inflammatory diseases, 1,500 to 2,000 milligrams day, under the guidance of a physician. 3. Vitamin B2, also known as riboflavin. It is a water-soluble vitamin and an indispensable coenzyme precursor for maintaining normal human metabolism. It is widely involved in energy metabolism, antioxidant reactions and cell growth and repair. It can reduce ROS and repair mitochondria. It is used to reduce the migration ability of lung cancer and liver cancer cells. Recommended dosage: general healthcare 100-400 mg day. Anti-inflammatory auxiliary 400-1200 mg day. Doctor's guidance is required. 4. Coenzyme Q10. Coenzyme Q10, referred to as COQ10, is a fat-soluble vitamin-like substance that is widely present in the mitochondria of human cells. It is a key factor in energy synthesis and antioxidant protection, especially in high-energy organs such as the heart, brain, and liver. It can protect mitochondria and inhibit inflammatory factors. Anti-migration evidence is a marker related, inhibiting breast cancer metastasis. Recommended dosage: general healthcare 100-200 mg day. Heart disease auxiliary 200-300 mg day. Doctor's guidance is required. 5. Resveratrol. It is a natural polyphenol compound widely found in grape skin, red wine, berries and peanuts. A large number of studies have shown that resveratrol has multiple biological activities, such as antioxidant, anti-inflammatory and anti-cancer. In cancer research, resveratrol is widely believed to have the potential to inhibit the spread and metastasis of cancer cells, mainly by regulating multiple signaling pathways. Recommended dosage: 500 mg day. Key points: curcumin plus omega-3 synergistically inhibit inflammation and metastasis. COQ10 plus vitamin B2 repair mitochondria and reduce cell escapability. Omega-3 and resveratrol inhibit EMT, MMP9, and inflammatory signaling pathways. Part 4. Research evidence of 5 natural anti-inflammatory factors in blocking the spread of cancer. 1. In 2024, Professor William Aronson and others from the University of California, Los Angeles, UCLA, conducted the CAPFISH-3 California Prostate Cancer Fish Oil Study 3 clinical trial. 100 low-risk prostate cancer patients were randomly assigned to a high omega-3, low omega-6 diet group, or a control group for 1 year. Research results: the Ki-67 proliferation index in the experimental group decreased by 15%, while the control group increased by 24%, indicating that omega-3 fatty acids may slow cancer cell proliferation and delay disease progression by regulating inflammatory responses. 2. In 2021, the National Cancer Institute, NCI, Barty et al. Evaluated the effect of curcumin on IL-6-induced STAT3 phosphorylation and NF-kappa B activation in multiple myeloma cells. Research results: curcumin effectively inhibited IL-6-induced STAT3 phosphorylation and NF-kappa B activation, and reduced the expression of cancer-related genes, such as BCL2, BCLXL, IL-6, and COX2. 3. In 2019, researchers from the University of California, Berkeley, explored the role of vitamin B2 in mitochondrial function and cancer prevention. Dose: 400 mg day. Results: high-dose vitamin B2 can reduce oxidative DNA damage and improve mitochondrial stability, thereby reducing the risk of mutation in precancerous cells. 4. In 1998, Dr. William V. Judy and others from the Southeast Biomedical Research Institute in the United States supplemented prostate cancer patients with high-dose coenzyme Q10 to observe its effects on prostate-specific antigen, PSA, levels and prostate volume. Supplement dose: 600 mg per day. Research results: within 180 days, PSA levels and prostate volume decreased by 73.6% and 48.4%, respectively, indicating that coenzyme Q10 may inhibit the spread of cancer by enhancing immune function. 5. In 2019, A Chinese research team used resveratrol to treat colorectal cancer, CRC, cells to evaluate its effects on cell invasiveness and related signaling pathways. Research results: resveratrol significantly inhibited TNF-alpha and TNF-beta-induced CRC cell invasiveness, reduced NF-kappa B activation, reduced the expression of oncogenes such as MMP9 and CXCR4, and regulated signaling factors associated with epithelial-mesenchymal transition, EMT. The study was published in the journal Cancer Cell International. These studies show that omega-3 fatty acids, curcumin, vitamin B2, coenzyme Q10 and resveratrol have potential anti-inflammatory and anti-metastatic effects in blocking the spread of cancer. However, the specific dosage and use of supplements should be adjusted according to individual circumstances and professional medical advice. Conclusion: we can't use nutrients to cure cancer, but we can take a scientific and firm step on the road to prevention and stopping its spread. Health is not just a state, but also a choice. Choosing anti-inflammatory means choosing a future that is farther away from cancer. Thank you for watching. Remember to like, follow, subscribe, and share this video to let more people know about these natural anti-cancer guardians.